Yes, our next guest is Liz Parrish. We should...
Now.
There we go. Now?
Yes.
Yes, our next guest is Liz Parrish, looking happy and radiant at 3 o'clock in the morning in Seattle. I don't know how she does it, but she does it. Liz is a founder and CEO of BioVita Science USA. It's a company creating gene therapies and other therapies to combat aging. She is an accomplished speaker and the happy subject of some controversy. She's presented at longevity conferences worldwide, and she'll be speaking today by video on the subject of gene therapies and advancing new treatments for longevity. I look forward to hearing from you, Liz. Thank you for coming.
Thank you, Liz. We are all ready to learn from you. Thank you.
Oh, thank you. Well, I am actually just trying to find my slides on the screen share because I want to share what we've been doing with you. Okay, here it is, I think. Hopefully these are the right ones. Okay, can you see my screen?
Yes.
Okay, well, good morning, and thank you so much for having me. It is a pleasure. It would have been more of a pleasure to be there with you. But as things are really busy on this side of the pond, as my grandma would say, I am not with you. I am across the ocean. But let's go ahead and get...
Can you put full screen? Full screen. Yeah, or a slideshow.
Yes, I will. Okay, can you see it now?
Perfect.
Okay, so yes, I think Paul said that we're controversial. I guess that is one way to put it. I think that we are right in time, and actually I don't think that we are controversial enough. I think we need to be moving a lot faster. My company has been working since 2015 in order to change your future outcome, change the way you age. And over the last few years, we really went back to the drawing board of what that meant. We knew already from my experiment in 2015, I took the first 2 gene therapies to treat biological aging. We knew that we needed a better delivery method. We needed more genes delivered to treat biological aging. That treating biological aging itself wouldn't be one gene because it's a complex disease. But not only that, we wanted to create the future that you wanted to see. We wanted to know, where do you want to go? And when we talk to people about what they really wanted for their life, they not only wanted optimal health, but they wanted to be able to break the mold genetically of what they were given. Some people wanted to do genius measures. They wanted to be the next Einstein. Other people wanted to live in extreme conditions. And many people wanted to just live to their absolute full potential and achieve what they'd like to achieve. And actually, this sounds off course, but it's all about treating your body genetically, treating biological aging, and creating an unlimited future. And it's not so far off as one would think. So what we wanted to do is we wanted to help you pack for your future. That's the way I like to say it, because we found what was wrong with gene therapy today is the most traditional viral vector, the one that I have taken now more than one time, has a very small payload. It's great in a lot of ways. It has a lot of benefit, although now we do know that it has a known toxicity at certain levels. And we were looking for a way in order to get multitude of genes into your cells that didn't have that limit in packaging size. We wanted to pack your suitcase full so that your future was delivered to you in one injection. So we went to the drawing board, and I think that probably a lot of people here have seen our BioRivix release. We will be releasing in journal. We have a supplemental piece of the paper that's coming out before we can do that. And that'll be exciting to all you researchers. But essentially what we did is we decided to go for a different viral vector. We didn't like the non-viral vectors because viral vectors are so awesome at what they do. And we wanted one in which we could pack a lot of payload in. We wanted to be able to get a multitude of genes because some people are born with limitations outside of the norm. They may have Tay-Sachs or cystic fibrosis. So we'd want to pack your longevity genes and your genes for your condition within the same treatment. We wanted an easier treatment, a treatment that would take less gene therapy in order to affect your body. And we also wanted to be able to redose it. And that's something that is very, very difficult to do today with the gene therapies they have. So we did we have a patent pending in CMV delivery, platform technology for a multitude of gene delivery. And what we had to do in order to verify whether this vector would work or not is we had to do some mouse studies. And probably everyone here knows that I'm not a huge fan of mouse studies, and you won't see me doing a magnitude of these, but I will do what needs to be done in order to prove therapeutics work. So what we did is we took 63 wild type mice. We treated them at 18 months. So they were already old mice. They were like, well, 56 to 62 in human chronological years. Not old, but where we would probably target humans right now. Then we essentially separated them into groups. We had the non-treated group. We had group treated just with the viral vector because we needed to make sure that it wasn't causing any problems. Then we had 4 other arms, both intraperitoneally, meaning within the stomach, and intranasally for follistatin and H-TERT. So a lot of people asked us, why did we do follistatin and H-TERT? There were already gene therapies around these. But when you're testing a new platform, you need something that's reproducible, and you want to work with something that you're looking for a known outcome. So there was a known outcome in these genes. And what we did, though, was even better than that. We tried redosing, which was fabulous. And here's some of the results. So what's statistically significant about this is that none of the treated mice died within the time of the controls. That's really important, you guys. And not only that, what was really a surprise was that the intranasal group did just as well as the group that was injected into the abdomen. And that simplifies gene therapy significantly. Let's move on. Lifespan. Our telomerase reverse transcriptase mice lived 41% longer than the controls. That very significant. The follistatin mice lived 32.5% longer. The TERT mice maintained their body weight, and the follistatin mice increased their body weight and kept a sustained increase in body weight across their lifespan. Glucose tolerance was significantly better in both groups, showing that these treatments affect metabolic health and could probably stave off things like type 2 diabetes and metabolic disorder. Glycated hemoglobin, much better. And physiological and physical condition, they were much faster at tasks. Repeated over and over again. I got to fly to New Jersey at Rutgers University where these mice were housed and actually meet them. They were all ladies, and they were quite fantastic. And one thing that I noticed is the follistatin mice dug everywhere. They were like super bodybuilder ladies, and they were going to dig everywhere. The telomerase mice were just very active, and the non-treated controls obviously already looked pretty rough by the time they were treated. But here are the mice at 24 months. So this is when the controls are starting to die. You can see the untreated mice in the upper left-hand corner. By the way, the mice that were treated just with the vector, they lived the same time as the controls. It did not have a detriment, which was great. You can see our follistatin ladies on the right. Were really big. And again, they were hilarious because they dug everywhere, whereas other mice don't bother with that. And then the telomerase reverse transcriptase mice are on the bottom left. And the lab helpers and the veterinarians likened them to 8-month-old mice for most of their lifespan. And they you can read the paper, their fur stayed thick and nice and much longer. And so it's actually quite fabulous. You can see them here at the same age. So the next steps that we'll be doing with this is we're actually going into human cell testing, and we have 6 drug candidates from this study, which is quite fabulous. Of course, we'll be doing combinatorial therapies as well. So what that means for you is a more awesome future. Now, when you're looking at aging, you're looking at something that seems kind of ubiquitous to most living species, and we have looked for ways to measure biological aging. And I'm really happy to say, you know, we dabbled in kits and we created a direct to consumer kit that has been shipping globally, and it's called the Timekeeper. And what we like about it is we looked at all of the open source data around 9 different papers, and then we created a proprietary algorithm that actually helped people assess their biological age based on their chronological age when they came in. So all of the clocks are not trained on the same age groups, and we were able to decipher where best to put that data. And what we like about this is it gives us some back-end understanding of what biological aging is. It helps people work on their interventions. Are your interventions working, or are they helping, or are they hurting you? So, you know, you certainly don't want to be spending money on something that isn't helping you. It helps you track your aging process. It's a bit of an educational piece, which I'm very much a proponent of. And it helps support critical drug development. You know, so a lot of people, they say why they choose our company is because we make drugs of the future, and they want to support that. So thank you for everyone who's done a Timekeeper and has stored their data in the BioKeeper. That's really fabulous. We have not launched this yet, but we are actually open sourcing gene therapy. We have over 2,400 entries of gene therapies that have either been in research or in human bodies, and we are open sourcing your future. Many of this actually even comes with the complete gene sequences. This is a project that I started with a PhD in Switzerland. She specializes in CRISPR, and she was fabulous. And it has moved to a student at Berkeley in the United States, and he has students that work with him to put it together and double check it. And I'm very excited about launching this project. This has not been launched yet, but from students, we've been told that as far as their academic careers, that this would have shaved 3 to 6 months off of their studies. So it's pretty fabulous. Now, Paul alluded to controversy. I think that it's really good controversy. First and foremost, I believe that humans need to partake in the future of medicine. Without human data, we can't move this technology forward. Our AI is becoming better and better, but without data, it really can't form an opinion on therapies. We partnered with several medical tourism companies, one of them called Integrative Health Systems, a few years ago. And we were lucky enough to have Maximum Life Foundation come forward and fund a study of 5 individuals participating in gene therapy. This was the first gene therapy in the world given to patients with dementia. It was overseen by our Director of Clinical Affairs, P.E. Sewell. And I'm going to run through his slides, some of his data here so that you can see. Usually he gives this presentation, or another doctor from Integrative Health Systems, but Jose wanted me to do this presentation. So here I go. Let's see if I can do it. So the treatment details, I should be able to do it. BioViva, my company, ran the data on this. And this is what we're committed to doing. We are committed to helping legitimize treatments, whether they work or whether they don't work. We need to know that now. And people are participating in these therapies, then we need to use that data for a better future. So this was delivered via a proprietary intranasal route. This is a patent pending that BioViva owns. It was an outpatient procedure and it only took 30 minutes. What is gene therapy? Gene therapy is a construct. It's a therapeutic gene that's packed inside of a what used to be a virus that can no longer get you sick. The viral genome is removed and the therapeutic gene is inserted. It can be built up to large scale, which has to happen in the case of gene therapy, and then it is administered. But the fabulous thing about this study is that it actually only used a very small amount of gene therapy. And that was actually a really big deal. We didn't plan on seeing the results that we did see from this study. My video is not playing, sorry about how gene therapy works, but essentially the gene therapy is delivered to the cell, it enters the cytoplasm, then it enters the nucleus, and it creates what's called an episome. So we do non-integrating gene therapies, and that way they don't, they're not subject to affecting your chromosome, and they're just considered safer. So in this cohort, there were 3 males and 2 females. I like to say that's a nice balance. Their chronological ages were from 72 to 85. They had mild to moderate dementia. And we did a multitude of data. So in order to work with our company, medical tourism companies have to step out of their comfort zone. They've got to do a lot of markers. There are timeframes in which these have to be done. They were developed in a very strong protocol. And the great thing is we get to look at a lot of biological markers around aging. And that's what kind of makes us different. We want to get to what's happening there. So one of the gene therapies that was used was telomerase reverse transcriptase. So why would we use something like this in the treatment of dementia? It's because when you repair the ends of the chromosomes, it actually has some benefit to the whole system of the organism. It improves mitochondrial function, which is something that we definitely see a lack of in dementia. And there is a great paper on how it reduces tau tangles. Improving the genomic stability of the cell is a really important part of better cell function. And that actually has benefits, again, back to the mitochondria. We believe that it reduces oncogenesis, although we will need to see that over a multitude of studies. We've definitely seen that in animal studies, and in model organism, it extends lifespan. So telomerase reverse transcriptase will be an incredible tool for increasing lifespan, getting more out of your life, not just a healthier life. The second gene that was used is called Klotho. So Klotho, there's a fabulous paper that came out actually after this study started. It was one of our hypotheses that it actually helps reduce beta amyloid plaques. It also repairs the myelin sheath, which is kind of like the insulation on the axon of nerves, helping them function better. It reduces oxidative stress. It restores microglial function. It increases cognitive function, which was a real pressing point in dementia. And in model organisms, it extends lifespan. So what did we see? We saw an increase in median telomere length. And I'm showing you this data. This is not something that we expected to see with such a small dose in a safety study. This, again, was a safety study with some efficacy. But in 4 out of 5 of the patients, we saw a significant increase in telomere lengths. And you'll see here, there's one that looks flat. Well, that is essentially like an increase because in 1 year they didn't age in their telomere lengths. So that's pretty cool. The 20th percentile telomeres got longer, and this is important because those short telomeres eventually become senescent cells, and those senescent cells, as you know, send off inflammatory markers. So in this, we were able to actually see an increase in telomere length in the shortest telomeres, which was fabulous. And then the biological age actually reduced. One person went up 1 year. They aged normally. We can see here, we always go, oh, too bad. But at least they didn't age significantly, as might be seen with dementia. And at the very top, you can see that someone, again, stayed their same chronological age over the period of aging 1 year. But then we also see some significant decreases upward of 6 years. Again, completely unexpected. The brain MRI results, there were no significant changes. You know, we will continue to actually ask those, ask integrative health systems to have their patients take more brain imaging to see if we can see something over time. But I think those were done at 10 months. There was no significant change. So a typical Fulstein test, a Fulstein test is where we saw our most significant benefits. This is what it looks like. It goes down over time. When you're diagnosed with dementia, you will generally lose 3 points every year. And this is just across the board seen. You can see here there was an intervention that they used in this scale, and it somewhat slowed the decline, but definitely still declining. But in our data, the patients from integrative health systems actually got better. And this is very significant. The paper has been written and it's being applied to journal. So we're really excited to get that out. It's not a cure. I think that there would be some ways to go, but it is a magnificent benefit and something that we hope to actually build on. So this suggests that hTERT and Klotho gene transfer therapy is effective at restoring cognitive function in mild and moderate neurodegenerative dementia. We're very excited about that. Further study with a larger population is warranted. Certainly, we will now need to go on and test this in a multitude of patients, and hopefully we can get support and funding from that. And this therapy could be applicable to general brain aging, increasing cognitive scores, affecting IQ in people who are on the earlier side of the spectrum of dementia. I want to thank David Kekich, whom hopefully will still be with us in the coming years. David's nonprofit, Maximum Life Foundation, was spearheaded this whole project. On BioViva side, we just analyzed the data. And I just, you know, he's one of my best friends. I talk to him every day before his recent cryonic storage. Okay, these are just coming through. You're going to see another paper coming through in the coming months, and that is what happens with telomeres and gene therapy in patient 0. That's me. My data was convoluted because I took gene therapy more than once. In case no one knew that. And here's what happened. So this is just the telomere data, and I just wanted to share some of it with you. It is not put in probably the most beautiful graphs at this point, but here's my average telomere length over time. You can see that it was going up, and actually my telomere length from the time that I took gene therapy until my last test, which was this year, has increased about 0.35 kilobytes per year. And that's pretty cool. I'm very proud of that. I'm very happy about that. So my percentile over time, I think I started in like the 20th or 30th percentile. My telomeres were critically short. I think my average age was around 60-ish something when I took my first telomere length before my first gene therapy. And it has now increased at a rate of 9.1% per year. This is my telomere length associated age over time. And since I took my gene therapy in 2015 and then another one in 2020, my biological age as associated with my telomere length has decreased by 5.3 years per year. So that's pretty fantastic. And no, I won't be Benjamin Button. And no, this is not every cell in my body. I so wish it was. But I'm really excited to share these results with you. And again, you're going to see a paper about a multitude of things that happened or didn't happen, things that stayed the same in my body over time. So I became so committed to ensuring that we get human data, that we actually start saving lives, which is the most ethical thing to do, that I decided to, over the last couple of years, go back to school and get my MBA. And my thesis, of course, is around investigating how the aging-associated non-communicable disease burden could benefit from emerging innovations in biotechnology via a medical tourism model. And you're going to see this come out as a white paper to the US government. I've created a new regulatory route in which would give people access to these type of treatments right here in the United States. And I hope that they take that into consideration. I have no idea where I'm at on time, but I want to thank you for listening. And if you have any questions, that's what I'm here for. Are you still
Liz,
there?
thank you for that. That was an excellent summary of what you've been doing, and it's very exciting. And it was timely. We actually have time for questions, which is highly unusual in an event like this. So
I always run short. If people give me 20 minutes, I do it like 10, but I thought for sure this one was going to be too long.
Right, well, it's the other.
Sorry about, but we're glad to have the time for you. So do we have any questions?
David,
yes.
Super talk, Liz. I want to ask about the treatment with telomerase for the brain conditions, which seemed very impressive. But I want to understand how this might work, because as I understand it, brain cells don't keep on dividing. Brain cells are for life. And so how would extending the telomeres assist in the brain? I can understand how it will assist elsewhere in the body.
There still is actually neurogenesis over time. There's a great study that we'll be watching that UC San Diego will be conducting in dementia, which is a gene therapy for BDNF, which will actually even increase that. So it is possible that some combination of these therapies will be beneficial. But telomerase and Klotho gene therapy are actually very beneficial for the cells that are static. So when you actually help them with genomic stability, when you lengthen telomeres, you create a better mitochondrial outcome. And Klotho helps with reducing the oxidative stress. So when you reduce the stress on these cells and you start to shore up some of the damage that happens even outside of cellular division, we plan to look at the minute data to see what's actually happening there. So these patients are definitely benefiting in cognitive function. They definitely had some telomere length extension, which we didn't expect. Remember, this was a small dose proprietary route intranasally. But what exactly is happening at the cellular level, that will take a lot of time to understand the mechanism. But these have been shown in other papers to have benefit in these areas of tau tangles, beta amyloid plaques, and cellular function, even in non-dividing cells.
Yeah, hello, Liz. I have one question. It will not surprise you, I think. So it's about maximal lifespan of the mice who were tested. Can you tell us precisely what was the maximal lifespan attained? And do you plan to have other tests on mice, maybe with already old mice, to see what's possible? And did you think also about testing for fertility? And I have, because I see David is still here, if David has also 30 seconds to answer. I like very much the video. You were speaking about gene therapies for diminishing suffering, and you were speaking about one woman, I think, who doesn't suffer. How do you know how far we are scientifically, let's say, in this direction?
Oh, that's questions. Hi, Didier. It's so nice to see you. I love your hair. You're fabulous. It's so good to see you and hear your voice. My kids always say, what happened to that guy with that voice that we used to hear on Sundays when we do the ILA meetings? So our mice lived over 40 months, and that is significant. So that is like the equivalent to, if your average age was going to be about 81, let's say you're going to die around 81, that would be to living over 110. So that is one question that I can answer to. Our next testing will be combining gene therapies and using them in human cells. And then, of course, we will probably go back and be forced to do some longevity study in mice, and hopefully we can break lifespans there. Of course, right now it is much focus on health span rather than directly to lifespan correlations, but we hope to get there and create those gene therapies that will actually bust the bottleneck of human lifespan.
And David's question.
Thank you, Didier. Yes, for the first time this year, the results were reported of intravenous gene therapy for an existing disorder, transthyretin amyloidosis, which is a lethal progressive disease. A one-off injection, a CRISPR infusion, actually seems, according to preliminary studies, have treated this, successfully treated the default protein, the faulty protein for this disorder. Now, exactly the same genetic intervention, one-off CRISPR infusion, could in principle be used not just for the recognized genetic diseases, but to give people everything from supercentenarian alleles to the very controversial alleles implicated in cognitive function, but more particularly, and this is my moral focus, in my little brief presentation, I mentioned Jo Cameron with her dual far and far out genes, which lead to extremely high levels of anandamide for bliss. In principle, though there would need to be trials first, one could use a one-off intravenous infusion to essentially give URI versions of the far and far out genes and elevate hedonic set points, tolerance of stress, and stuff like this. There are horrendous pitfalls, which I'm glossing over in the context of a presentation stroke replies to a question. But yes, essentially the biotech revolution, biohappiness revolution, is going to embrace not just future children and grandchildren, but can in principle help existing people.
Oh, yeah. And I mean, to build on what David is saying, there are 5 regulated, passed, and approved gene therapies to treat disease. So, severe combined immune deficiency has now been treated with gene therapy, lipoprotein lipase disorder, spinal muscular atrophy, and others. Sickle cell anemia was just treated in the last 2 years with CRISPR technology. It's an ex vivo gene therapy that treats the cells in the bone marrow, and these patients are now not having sickle cell disorder during activity. So this is an emerging technology, but it's not, just a future technology. This is already being used in the bodies of people for functional cures. So it's good to keep in mind that we are in the gene therapy space coming far out of the proof of concept stage. As far as treating biological aging, we have a ways to go.
Excellent.
Thank you so much, Liz. This was certainly informative. I'm afraid we have time for no more questions, but we do have a word from our producer, José Cordeiro, who needs to talk about logistics. Liz, thank you again. You should probably make the best of the rest of your evening or morning or whatever. But we're very happy to have you here as always.
Yes, thank you so much, Liz and David. And we continue here in Madrid. Live long and prosper, my friends.